Welcome, guys. So this will be the final module that I'll be covering for this course. So this one's going to be another case study like our biosignal processing one, but this time we're going to do a model of the cardiovascular system. And just like the biosignal one before, this makes heavy use of the previous module. So in this case, it was the numerical methods. So if you need a quick refresher on that, go ahead and rewatch that video, go over that PowerPoint, because you're going to be using some of the skills we learned there in this module. So naturally, we can use computational software like Matlab to make mathematical models, and we can use them to describe physiological systems and processes. And then once we've described them mathematically, we can go ahead and analyze how the human body works under various disease states that you wouldn't be able to readily do on actual patients in real life, but you could easily do it inside of our model. So that's what we're going to do in this module. We're going to make a very rudimentary Matlab model for the cardiovascular system, and then you're going to use that model in the quizzes to answer some questions about disease states and how the human body works depending upon where blood flows and how much it flows here and there. And it's going to be very interesting and exciting, assuming you can get through all of this. Now, first off, we're going to have to do a quick little introduction on anatomy of the circulation. So I realize you're probably taking this as a freshman, and you may or may not have ever had any introduction to the anatomy of the circulation system in the human body, even if you were taking biology in high school. So to sort of cut that off, this might be your first foray into how the circulation system works. So I'm going to go a bit slow through this and try to explain each individual part. It's really just a giant loop that repeats over and over again, but once you have the loop down, it sort of becomes second nature to you. But if you already know all this, you can go ahead and skip to the next part of the video where I actually explain how do we model this complicated little system we got here. If not, then join me as I try to explain what a biology teacher would do. So the components of circulation are generally just three. So you have the heart, which does the main pumping of the blood. You have the systemic circulation, which is where you have blood going to all different parts of your body. And then you have the pulmonary circulation, which refers to the part of the circulation where blood goes to your lungs to get reoxygenated. And if you see here in this little figure, you have these percentages here. The percentages represent where blood spends most of the time in your body. So you can see here, arteries 13%, arterioles and capillaries 7%, veins and venules 64%, heart 7%, pulmonary circulation 9%. Generally, most of the circulation in the body is happening in the systemic vessels. Some of it is in the pulmonary circulation, and some of it is in the heart, but most of it is in systemic vessels. So first off, let's talk about the heart. So the heart consists of two sides, and as counterintuitive as it sounds, the two sides are left and right, and over here, the right side that you're looking at is actually the left, and this side in blue is actually the right, mainly because of perspective of how we're looking at it. Don't worry, you'll quickly get over it. There are four chambers of the heart, so we call them the left atrium, left ventricle, right atrium, right ventricle, and they're located right here. So you have the left atrium, left ventricle, right atrium, right ventricle, and along with these four chambers, you also have the four valves. So you have two on the left side, two on the right side. They're called the mitral, aortic, tricuspid, and pulmonary. They also have more medically used names called atrioventricular, semilunar, atrioventricular, and semilunar, though I think there's an extra bit to these names I can't remember, but I usually just call them mitral, aortic, tricuspid, and pulmonary, and they're located in between each of these chambers. So first, let me talk a bit about how these four valves work, and then once I go through each of the individual components, we'll do a quick loop on how the blood flows, and hopefully it ties everything together nicely. So first off, about the valves. So the valves beat in synchronized type of fashion. So when the heart contracts, we call that systole, and blood exits the left and right ventricle through the aortic and pulmonary valve at the exact same time. So that would be up here and over here. So pulmonary valve, aortic valve. The ventricles contract and they push the blood into the lungs and throughout the body at the exact same times. Then when the heart relaxes, we call that diastole, and the left and right ventricles start filling up with blood in preparation to pump, and the atrium start dumping blood into them. And those occur because the mitral and tricuspid valves are open, and those are found here and here. So these two valves, the pulmonary and the aortic, are closed, and the tricuspid and the mitral valves are open, and so now the left and right ventricles start filling up with blood, and then these close, and then the pulmonary and aortic valves open, and then blood gets released again. So they always go in fashion, one after the other. So here's a quick simplified model of the heart. So you have the major chambers, the right atrium, right ventricle, left atrium, left ventricle, as well as the 4 valves: tricuspid valve, pulmonary valve, mitral valve, and aortic valve. And then one part of this loop, we call it the pulmonary circulation, which goes by the lungs. And the other part of the loop, we call it the systemic circulation, that goes through the body. And this little summary basically just shows you how the blood flows through the heart, but in words. I don't find it that useful, but you can read this. I'm going to go ahead of this and then explain the loop after I talk about each individual circulations. So you can go through this as like sort of a summary. So first off, the systemic circulation. So the point of systemic circulation is to deliver oxygen and nutrients to different parts of your tissue. So once blood leaves the heart, it goes through the very large artery, which we call the aorta, and then the aorta starts branching into smaller arteries, and those branch down into very even smaller arteries, which we call arterioles, and those branch into what are called capillaries. Now it's at the capillaries where oxygen exchange, carbon dioxide exchange, nutrients exchange happen with our cells, and then once that's done, the capillaries join together, and they form the very small venules. Those join together to create veins. Those join together to create what's called a gigantic vein called the vena cava, and that goes back to the heart ready for reoxygenation. And the only reason why we color these red and blue is just for sort of picturesque purposes. Blood is still red throughout this entire loop, but it's easier to show deoxygenated blood as blue and oxygenated blood as red. So now that the blood has entered back into the heart from the vena cava, it's deoxygenated, so now it has to enter the pulmonary circulation to get reoxygenated. And that occurs through the same sort of system. So you have, once blood exits the right ventricle, it goes into the pulmonary circulation through pulmonary arteries. Those go to the lungs, both of them, down all the way to capillaries, and these capillaries go right up against what are called alveoli. And those alveoli allow for oxygen to enter into the blood, carbon dioxide to exit, so you can breathe them out. So that's this exchange that's going on right here. Then naturally those capillaries join together, create veins. Those veins join together, create venules, and then that goes all the way back into the heart, that into the left atrium, then into the left ventricle, and then the heart just pumps one more time and pumps the blood right back out. And here's a little video that you can go to that sort of explains this loop. You can if you want. I'm not going to show it to you. And here's a grossly oversimplified version of the heart, where they basically simplified it into a closed loop system that has 2 sides, left and right, and 2 circulations, systemic and pulmonary. So they have a right-sided heart, left-sided heart, systemic part of the loop, pulmonary part of the loop. And then here are the individual components of the heart and how the blood sort of just goes in a loop around them. So now I'm going to go back up here, and like I promised, I'll explain the loop as it goes. So I find this figure to be the best one to explain the loop. So let's assume the blood has just finished oxygenating every single part of your body, and it's going back to the heart and getting prepared for reoxygenation. So first off, blood enters through what's called the superior and inferior vena cava, which is a really large vein. So over here is demonstrated by the blood coming in from the upper body and the lower body. And so we call the superior vena cava and inferior vena cava. So the blood pools together into what's called the right atrium, and then the blood enters in through the the tricuspid valve. So now the blood enters in through the tricuspid valve. It starts filling up the right ventricle. Then when the right ventricle is filled up, the blood exits through the pulmonary valve, and because it's called pulmonary, that means it's going to the lungs. So then the blood enters the lungs, then it goes into the pulmonary circulation, gets oxygenated, and then it comes back through the pulmonary veins into the left atrium, pulls up into here, then it goes through the mitral valve, pulls up in the left ventricle, then the left ventricle does one final push and pushes the blood that's now oxygenated through the aortic valve. That goes up towards the aorta, the largest artery you have in the body, and then it exits. Some of it goes up to your head and upper limbs to oxygenate your brain and your arms and such. Then the remainder goes all the way down to your lower limbs and lower part of your body. Then that goes through the systemic circulation, goes through your cells, arterioles, venules, capillaries, then it joins back together to veins, and then that comes right back here to the inferior and superior vena cava, ready to be reoxygenated again. And that is basically it. This loop just repeats over and over again, 60 times a second, if I believe, right? Heartbeat 69 seconds. it will be more or less depending on how much of an athlete you are. So some athletes, their hearts beat way slower than usual because their heart doesn't have to beat as quickly. For others, you can have a very fast heartbeat. So this loop might be going many times a second for you, or many times a minute for you. But generally, that's a gross overview of how the loop works in terms of biology. So join me in the next video where I actually explain how we're going to model all of this. That should be fun. See you guys then. Alright, so how do we actually go about modeling this complicated circulatory system with even in our simplified sketch where we only have the pulmonary circulation, the systemic circulation, left, right heart? How do we actually model it mathematically? Well, we need to make some assumptions naturally. So the first thing we need to ask ourselves is, do we model blood vessels as a pipe or a balloon? So let me explain what I'm talking about here. So small blood vessels, meaning the arterioles, the capillaries, the venules, they actually have very significant resistance to flow. So imagine water flowing through a very large pipe versus it moving through something with a very narrow and small diameter. It takes much more force to get the water to flow through a very small hole than it is to get it to flow through a very large opening. So that's why we could say small blood vessels have very high resistance to flows. And because of that, it's sort of the pressure drop between where the blood enters and where it exits. That pressure drop between those two spots is a lot like water flowing through a very rigid plumbing pipe. And conversely, if you have a very large vessel, so you have the aorta, the large arteries and veins, the vena cava, pulmonary arteries and veins, the very large blood vessels in comparison to very small little capillaries, those actually behave more like what we call balloons, where they can inflate and contract to accommodate all the blood that's pouring into them. So they actually increase in volume as blood pressure increases. So one is like a very rigid pipe for small vessels. The other is for very large blood vessels, and it behaves more like a balloon. And the heart itself, the left and right side of the heart, we're going to treat it as a pump because it's not really a pipe. It doesn't transfer the blood. It's not really a balloon. It doesn't just inflate. It's a pump because it actually contracts and pumps the blood throughout the rest of the body. So those are the three things we're going to try to model. So here's a quick breakdown. So you have the pulmonary circulation, then the blood enters the heart, and in this case, from the pulmonary circulation, you have, well, I guess we can start from one. So starting here on the left side of the heart, we have it as a pump. So this is the left atrium and ventricle. Then it goes into the systemic arteries. Then it goes down to in this case, the systemic arterioles, the very small vessels. And because they're small, we can model them as pipes. Then they join together into veins, and these are systemic veins. Then it goes into the left side of the heart, the right side of the heart. The right side of the heart pumps the blood, goes into the pulmonary arteries. That goes into the pulmonary arterioles, capillaries, venules, so the pulmonary circulation, which is also small and rigid, so we have that as pipes as well, collect into the pulmonary veins, which we're modeling as a large vessel as a balloon, and goes right back to the left side of the heart, and the cycle repeats again. Balloon, pump, balloon, pipe, balloon, pump, balloon, pipe, and keeps going like that. So here are some labels for them: pulmonary veins, left heart, systemic arteries, all the other vessels of systemic circulation, systemic veins, right heart, pulmonary arteries, pulmonary circulation, and the loop just continues onward. So first, how we're going to model these balloons, which are our large vessels. So like I said, they have a relationship between volume and pressure. So the units for volume are going to be in liters. The units for pressure are going to be in what's called millimeters of mercury, which you might remember from, I believe it's chemistry, where you first learned about it. So the relationship between volume and pressure in blood vessels is mostly linear. It has some non-linearities to it, but you can generally call it approximately linear. And so if it's linear, that means that it has a slope of some kind, which we're going to call c. So this will be in our y equals mx plus b classical equation. This will be our m. And it also has a y-intercept. So instead of it being a b, we call it v0, which would be right about here. And that's when the residual volume, and we call the intercept v0 is a residual volume when pressure is 0. Now some of you, well, let me first explain the equation. So the following equation is how you describe it. So the volume you have is equal to whatever dead volume you have, or residual volume, plus some compliance times pressure. Y equals mx plus b. Now you might be wondering, why does the blood vessel have a y-intercept at all? If you have no pressure in the blood vessel, shouldn't you just have no volume at all? Not necessarily. Imagine you're breathing in and out. Even if you were to breathe out all of the air from your lungs, there's still a little bit of volume left. That's dead volume. So similarly, if you empty out your blood vessels of all of their blood and there's no pressure exerted on any of their walls, there's still some small amount of volume in there. We call that residual volume. And even if it's a very small value, it's still there, so we still have to account for it. So the next couple of slides go over how do we use this equation to figure out what are the various parameters we need to model these 4 balloons. So I'll just show you the examples for them. So I'm not going to actually do them because you have to do them yourself, but they use heavily the methods we learned from Module 7. So here's what we're going to do. So for the systemic arteries, systemic veins, pulmonary arteries, pulmonary veins, those 4 balloons, we have pressure and volume information. So 50 millimeters mercury, 70, 90, 100, and so forth. And the volume, so 1.28, 1.54, and so on and so forth. So if you go back to the previous module where we were doing numerical methods, and you plot both of these together, you can fit a linear equation to this data set and figure out in a linear equation what is our V0 and what is our C, which I taught you how to do in the last module. So just go into Matlab, plot these against each other. Then you can either use the figure basic fitting tools, or you can use one of the handy interpolation functions or fitting functions and figure out what VSAO are and CSA. And if you forget, just imagine like this is X and this is Y, and you can use one of the examples I did in the previous module. Likewise, you can do the exact same thing for the systemic veins. You can do the exact same thing for the pulmonary arteries. Just imagine this is X and this is Y. And then you can do the exact same thing for the pulmonary veins. X, Y, figure out after you fitted what VP VO0 and CP V are. So this is your slope, this is your y-intercept. Once you figure those out, that's pretty much it for modeling the balloons. So that's for the large arteries. Now what about the smaller ones? So for small vessels, remember how I said we're gonna treat them like a pipe? So how would you measure what's going on in a pipe? So the only thing we know for certain is when the pressure goes into the pipe on one end, so let's say the pressure in the systemic arteries goes in one end, the pressure that comes out in the systemic veins is going to be much less. So the heart pumps with a huge amount of pressure that enters into systemic arteries, but then by the time the blood has gone through all parts of your system and it's pulling together in your veins, the pressure is really low because it's met with a lot of resistance along the way. So that a way that we can quantify how much resistance is going through this from one end to the other is to look for something that stayed constant. And what stayed constant is this variable we're going to call Q, or this parameter really. So what is Q? Q is the flow. So assuming that you're not bleeding anywhere, the flow that enters in through your systemic arteries is the same flow that exits through your systemic veins. So if that stays the same, that means that the difference in pressure, so pressure in minus pressure out, divided by some sort of resistance value, should be a constant amount of flow. And this RS is going to be sort of a lumped together approximation of how much resistance there is through all these other vessels. And this is going to be the explanation between how you have high pressure here and low pressure here. So similarly, for the pulmonary circulation, instead of it being from the systemic arteries minus the systemic veins, we have it be the pulmonary arteries minus the pulmonary veins divided by what we're going to call RP, which is like RS, but instead of it being the systemic resistance, this is going to be the pulmonary resistance. And it looks like Dr. Sukis was nice enough to give you guys what RP was, which is 1.79 millimeters mercury divided by liters divided by minute. And if you're wondering, oh, now what's RS? Ta-da, next part of the thing. So this is the part where you're going to have to work a little. So you're going to have to estimate what the systemic resistance RS is using some information I'm about to tell you. So first off, the resistance for a flow through a cylindrical pipe, like a blood vessel, can be estimated using what we call in fluid dynamics Poiseuille's law, and it's this equation right here. So 8 times n, although we usually call that nu, times L divided by pi r to the fourth, where n is your blood viscosity. So that is blood viscosity is a measurement of how easily blood flows. L is the length of your vessel, r is the radius, and knowing these parameters, you can figure out how much resistance you have through a particular pipe. In our case, our pipe are our blood vessels. And Dr. Suki has already worked out an example here for a single capillary. So you might be wondering, where in the world is he getting these numbers from? That comes from this slide here. So it might be easier if I explain this slide here and then jump back over here. So The following data from Roy and CCOM, these two guys are big names in the microvascular modeling field. They provide some dimensions, radius and length, and blood viscosity for different vessels, arterioles, and venules for the systemic circuit. And what we want you to do is, can you estimate the resistance for each of these vessels? So in the systemic circulation, remember you start off with the systemic arteries, and then it goes to the systemic veins. So we're gonna expand that to have what we're calling the upstream vessels. Then they lead into what we're calling feeding arteries. Those branch into arterioles, more arterioles, more arterioles. We'll call them first, second, third order arterioles. Finally, we have terminal arterioles, and those finally branch out into capillaries. And then those capillaries join back together into veins or venules. Then those venules join together to create a draining vein. Then this becomes a very large vena cava and then goes back up to the heart. So we basically just expanded the systemic arteries, systemic veins to have all these extra components to them. So you might be wondering, why do we just complicate our life? Because each of these little components, this upstream vessels, feeding arterioles, first order, second order, third order, and so forth, each of them have a contribution to the total systemic resistance of our circulatory system, and we need to actually figure out what that is. So you can see here a nice little table that has the upstream vessels, the feeding arterioles labeled accordingly, all the way back to the veins, and we have the radius, the length, the viscosity, and the number of vessels. All of these are parameters that we can use in Poiseuille's law to figure out what the resistance is. And I'll show you a quick workup on the capillaries. So, using this equation, we know that the length for capillaries from this table is 0.0545 centimeters. The radius is 0.0003 centimeters. The blood viscosity is 0.0905 poise, which is a fancy way of saying grams per centimeter per second. And if you use these values, which are the exact same values from this table here, you can figure out that the resistance is 1.55 times 10 to the 12th, and this mess of units right here. So this is not the way that it's reported down here. The way it's reported is in the units of millimeters mercury per liter per minute. So to do that, you're gonna have to convert. And you can look at the way that it's being converted, but it's a bit just messy dimensional analysis. Looks like you're dividing by 10, dividing by 133.322, multiplying by 1,000. What this boils down to is you take whatever your number you have here, multiply it by 0.0125 millimeters mercury per liter per minute, and your output answer is in the units that you want. So it looks like the total resistance of the capillary system is 1.937 times 10 to the 10, which is this exact same value. So go ahead and do that for each one of these combinations. I would suggest you write a little Matlab script that basically just takes in a variable of R, L, N, and N, and then just spits out what the resistance is, because I doubt you guys want to do this on a calculator. So that is the resistance for the entire capillaries, all the capillaries together. But what if we want to just have it per vessel? Well, we need to divide it by however many vessels there are. So because there are a large number of vessels, you need, specifically, it looks like for the systemic capillaries, there's 6.5 billion of them. We can take the total resistance, RC, which we just calculated, and reduce that to a single capillary by dividing everything by dividing the total by however many vessels we have. So we can turn this very large 1.937 times 10 to the 10 divided by 6.47 times 10 to the 9, and we get that each of the capillaries individually have a resistance of about 2.99 millimeters mercury per liter per minute. So assuming you have all of these resistances from the knowing what the radius, length, and viscosity and number of vessels are, you can now figure out what the order is. So basically just use these first three numbers to figure out what the resistance is in total, then divide it by this value right here, and you can get the smaller resistances here. And this is more of something you'll learn in circuits, but if you have these different orders connected in series, so one connects to the next, connects to the next, and so on and so forth, the total resistance across all these vessels can be said to be the combination of just you adding all of them together. So if you just add what this resistance is, what this resistance is, what this resistance is, so on and so forth, all the way through, that will be the total resistance in systemic circulation, and that will be RS. So once you fill out this table, just sum together what the resistances are, and you can put that right here. And that will be the single value that you're going to be using to account for the blood flow through the systemic circulation. And you should get a value something closer to this in terms of magnitude, so you can sort of use this as a reference. They might be different, but as long as you're not like 10 to the 9th or something, you can use this as something to compare it with. All right, so that's how to model the small vessels, large vessels. Now let's talk about how we're going to model the actual heart itself. So we said the heart is like a pump. So the volumetric flow of blood, calling that Q, that each side of the heart pumps is proportional to the blood pressure of the veins supplying that side of the heart. So we're going to use a very simple equation where we're going to say, okay, depending on how much pressure is entering the heart, if you multiply that pressure by some sort of constant, we're going to call the pump coefficient, that should stay constant, and we're going to say that should stay constant and call it Q. And this should be the same for the left and right side of the heart, but the constant that is being used changes. So the pump coefficient is a fancy little constant that you can actually approximate by using the heart's beating frequency and the left heart's compliance at the end of diastole to figure out what it is. So specifically, this KL can be found out by doing the frequency times compliance during diastole. So F times C should figure it out. So blood enters into the pulmonary veins, and then if you want to figure out what the pressure is, if you want to figure out what the pump coefficient is, you just take the 70 heartbeats per minute, then you multiply by the C diastole, and that should give you what this coefficient should be. So you can go ahead and do that, and you should get some sort of value here. Likewise, for the right side of the heart, in this case, the blood is entering in, so the pressure is coming in from the stomach veins. So the pressure here is from the stomach veins, and for the right heart pump coefficient, you still use the same F, but now the C changes to 0.004 liters per millimeters mercury. Multiply those two values, and you should get what the right heart pump coefficient is, and that should do it for the left and right pumps. And now is when we bring everything together, and I think I'll save this for the next part of the video. So see you guys then. So now that we have all of our equations, let's see how we're going to integrate them all into a single model. So remember, here's our simplified model, and we have the equations for our balloons, our smaller vessels, which are pipes, and a left-right side heart. So the variables that we don't know are the pressures and the volumes. So we have 2 for the pulmonary veins, 2 for stomach arteries, 2 for stomach veins, 2 for the pulmonary arteries. I think I said that twice. We also don't know what the flow is, and that is essentially it. Everything else in our model is a constant that we figured out using either some quick multiplication or doing some sort of annoying multiplication here, using some known equation, Poisson's law, or we figured them out doing some fits using some known data. But everything else is a variable that we don't know. So how do we solve for that? Well, if you remember, we have 8 equations, but we have 9 unknown variables, 2, 4, 6, 8, 9. So we need one more equation. And what's that last equation? Well, we can make an assumption that the blood volume is conserved. Why? Well, unless the person we're trying to model is losing blood actively, you can be pretty rest assured and confident that when you add up together the volume that's in the 4 parts of the heart, so the pulmonary arteries, pulmonary veins, systemic arteries, systemic veins, if you add together all these volumes, the total volume should stay constant for your subject. So we're going to say the total volume of all of these should be 5 liters, which is a healthy estimate for a normal human adult. So now we have 9 equations, 9 unknowns. That sounds like a system of equations that we can actually solve now. So ignore this, and I think this slide is more easier to explain. So now that we have our system of equations, so here are 9 equations, we can actually use the methods from Module 7. So, for example, the Gauss elimination method, or doing inverse A times B and such to solve this system of equations. But we can't just plop these all into Matlab and then just throw them in and then just solve. We need to prepare these equations a little. So how do we do that? Well, remember, you want all of your variables to be on one side and all your constants to be on the other side. So let's take, for example, the first line here. So this first equation for the systemic arteries, you have the volume of systemic arteries is equal to some residual volume plus some compliance constant times the pressure. So volume and pressure are our variables, so we're going to move those to the left side, and we're going to move everything we do know to the right side. So the only part of this entire equation we know is the volume. We also know what CSA is, so CSA is sort of attached to this PSA, so consider this as just part of the variable. So we're going to move VSA and PSA to the left side, and we're going to keep VSAO, which we know, to the right side. So one side to the left, the other side to the right, and everything on the right from all these equations are going to be things that we know for sure. So they're either going to be constants, so VSO, VSAO, VPO. So there's a slight error here. This should say VSV. So this should say VSVO, VPA, VPV, 000, and the total volume. And everything on the left side should just be constants times a variable of some kind. So now that we have left side as variables, right side as constants, we can actually go ahead and put it into a very nice large matrix. That's going to be, because we have 9 variables and 9 unknowns, a 9 by 9. So how do we do this? So if you want to put the first variable, let's say, each column of our matrix is going to be dedicated to one of our variables in our model. So just to make things easy, let's just have it be in order. So we have the volume of systemic arteries, then volume of systemic veins, volume of pulmonary arteries, pulmonary veins, the flow, and then the various pressures. So the very first line of our matrix, everything that for our equation is in our equation, we're going to put it here. Everything that isn't, we're going to leave it as a 0. So in this case, VSA and PSA exist in the first equation, so we're going to put those here and here. And naturally, because this PSA is going to be multiplied by CSA, that comes along for the ride as a negative, and then it's just going to be a bunch of zeros, and then it's going to be equal to whatever it is here on the right. So VSA is L0. And for example, if you want to do it for, let's say, this equation right here for the left side, I believe, of the heart, what you can do is notice that the Q has an RS next to it, so you have 1 over here, and you have it being multiplied by an RS, and then you have this PSA variable also existing, so this would be a negative PSA here. Then you have this PSV existing here, and so this would also be, but we don't have any of the other variables inside of this equation, so all of these are just zeros, and then it's going to be equal to whatever the right side is. So when you write it down, here's what it's going to look like. Everywhere you see a 1, that means that equation had that variable in it. So back up here, VSA was inside of this equation, so it was in the very first spot and it exists, so we're going to put a 1 here. But VSV, VPA, VPV, Q, SV for the pressures, none of those exist in our model, so those are all zeros. Or none of those exist in the first equation, so all of those are 0. Except for the pressure in the stomach arteries, which does exist. So we're going to have that be a negative CSA here. And then this is going to be equal to VSAO. And you just go equation by equation, filling out this entire matrix, and you should just end up with a 9 by 9 matrix. And when you get this giant matrix, remember that if you consider this A, and you consider your output B, what you want to solve for is AX is equal to B, and you can use the methods we talked about in our previous module, so Gauss elimination, left division and such, to figure out what X is. And remember, X is all of our unknowns. So the order we specified the variables up here, so VSA, VSV, VPV, VPA, VPV, Q, and so on and so forth, that is the same order that your X vector is going to come out to showing you when you solve this matrix in Matlab. Naturally, I'm not going to do it for you, but hopefully I've explained a little enough so you can do it on your own. And if you need a quick refresher on how you actually solve some more simpler system equations, go back to the last module where we did some examples on, I believe it was system of 3 equations. So if you can do a system of 3 equations, you can easily do a system of 9 equations, considering how I already explained how to put each of the components in. So I think that does it. It's been a pleasure making these videos for you guys, and hopefully you've learned something. If you see me around the apartment, come by and say hi. Bye.